Okay, so good afternoon, students. I welcome you to the problem solving session and the tutorial session for the contents of week 5 of this NPTEL course, Introduction to Cell Biology. So, as we have done in the previous classes, in this session as well, I will first run you through the summary of the content that has been taught in this 5th week of this course. And you can stop me if you have questions at any point of time. And also at the end, we can discuss questions regarding to week 5 or even the previous week's content if you have any. Okay, so in this 5th week of the course, Introduction to Cell Biology, the primary topics of discussion had been the structure of the cell, in which the instructors have taught you the structures of the cell wall, the cell membrane, and the cytoskeletal elements, right? So, cell wall is a component which is seen in both the prokaryotic cells, that is in the bacterial cells, as well as in the eukaryotic cells, that is the plant cells. So, in case of the bacterial cells, there is the cell membrane or the plasma membrane, and outside of the plasma membrane, there are two layers. One is a peptidoglycan layer, which is a cell wall. The cell wall, which is primarily made up of the peptidoglycan layer, and there is a second lipid bilayer, which is quite distinct in the lipid composition, which is called the outer membrane. And together, in case of these bacterial or prokaryotic cells, the cell membrane, the cell wall, and the outer membrane together is referred to as a cell envelope. Whereas in case of these plant cells, which are eukaryotic cells, there you have the plasma membrane or the cell membrane, and outside the plasma membrane or the cell membrane, you have again cell wall, which is made up of primarily polysaccharides like cellulose, hemicellulose, and pectin. And that also can be divided into two layers basically. One is a primary cell wall and the other is a secondary cell wall. Now, the cell wall is actually a very tough layer made up of these polysaccharides, peptidoglycan in case of the bacterial cells, and this cellulose, hemicellulose, and pectin in case of the plant cells, which primarily provides mechanical support and rigidity to both the bacterial cells as well as the plant cells. It has different functions to a certain extent in case of bacterial cells versus plant cells. For example, the mechanical support and rigidity that the plant cell walls provide also becomes useful for the plants to grow against gravity, right? Where the wood comes in very handy, this mechanical strength and rigidity, where they grow so tall against gravity. So this cell wall also helps in that aspect in case of the plant cells. But both plant cells as well as bacterial cells, the cell wall also, this mechanical support that the cell wall provides also protects the cells against hyperosmotic stress, where there can be water influx into the cells can swell, and there can be a danger of lysis of the bacterial cells or plant cells in the absence of the cell wall. So that's how cell wall is a cell wall made up of these polysaccharides, be it peptidoglycan in case of the bacterial cells or cellulose and pectin in case of the plant cells, forms a rigid, tough, but also has some flexibility, and also it has some porous nature, so the molecules can, the very small molecules can actually, small molecules or proteins or signaling molecules which are necessary for the crosstalk, the communication, cell to cell communication, both in case of plant cells as well as in case of bacterial cells, like quorum sensing in case of bacterial cells, can happen. But they provide primarily mechanical support to the cells, to the cell membrane, and they provide protection against the hyperosmotic stress, helps in preventing the cellular lysis in face of hyperosmotic stress. Now let's look at bacterial cells and the bacterial cell wall and the plant cell wall in a bit more details, and the molecular composition of the bacterial cell wall and the plant cell wall, right? So the bacterial cell wall, bacterial cell wall can be of can have two different architectures depending on the type of the bacteria, right? So actually, depending on the type of the cell envelope of the bacterial cells, the bacterial cells are classified into two different categories. One are called the gram-positive bacteria and one is the gram-negative bacteria. This classification arose because of the differential responses of different bacterial cells to the gram staining protocol, which we will discuss in one later slide. So this differential response of bacterial cells to the same gram staining protocol was found to be because of the different architecture of the bacterial cell envelope. So the gram-positive bacteria, in case of the gram-positive bacterial cells, they have the cell membrane or plasma membrane, and outside the plasma membrane, they have the peptidoglycan cell wall. They have a thick peptidoglycan cell wall, whereas the gram-negative bacteria, they have the cell membrane or the plasma membrane, and outside this plasma membrane, they have a much thinner peptidoglycan layer. And because of that, this space between the plasma membrane and the peptidoglycan, which is called the periplasmic space, they have a quite extensive periplasmic space as opposed to the gram-positive bacteria. But in case of gram-negative bacteria, the most important thing is that outside this thin peptid, relatively much thinner peptidoglycan layer as compared to the gram-positive bacteria, they have an extra layer of lipid bilayer, which is called the outer membrane. However, the outer membrane is very distinct in the lipid composition. Several very unique lipids are present in the outer membrane, which are quite different from that of the plasma membrane, like lipid A, which are also major antigens that are recognized by our immune system when these bacterial, many of these gram-negative pathogenic bacterial cells invade our bodies, invade different tissues and organs of our bodies, basically. Okay, so this peptidoglycan actually is peptidoglycan is actually a polysaccharide. Peptidoglycan um, of the bacterial cell wall, gram-positive bacterial cell wall as a gram-negative bacterial cell wall, consists of long fibers of polysaccharides, um, which are formed because of polymerization of the monomeric sugar acids like N-acetyl muramic acid and N-acetyl glucosamine. So this monomeric sugar units like N-acetyl glucosamine and N-acetyl muramic acid polymerize because of formation of the glycosidic bonds to form long polysaccharide chains. And then these polysaccharide chains, from these polysaccharide chains, actually from the muramic acid residues of the polysaccharide chain, short 5 amino acid long peptide chains come out. These short peptide chains consisting of 5 amino acid residues are joined to this N-acetyl muramic acid residues of each polysaccharide chain. And then the adjacent polysaccharide chains are cross-linked together because of formation of peptide linkages between these short polypeptide chains that are coming out of the polysaccharide chains made up of N-acetyl glucosamine and N-acetyl muramic acid. That's how, because of this cross-linking of the polysaccharide fibers or long polysaccharide chains made up of N-acetyl glucosamine and N-acetyl muramic acid, this peptidoglycan cell wall of the bacterial cell envelope is formed, which also you can see very nicely in this slide. For example, um, each of these are a polysaccharide fibril, right, which are made up of as a result of the polymerization of these monomeric sugar residues, sugar amines and sugar acids like N-acetyl glucosamine and N-acetyl muramic acid are joined together because of formation of glycosidic linkages, glycosidic bonds. Now from each of these polysaccharide chains, you can see the short polypeptide side chains, which are made up of like 5 amino acid residues, are coming out, and then the adjacent polysaccharide chains are cross-linked because by formation of these peptide linkages between these polypeptide chains consisting of short polypeptide chains that are coming up, leading to cross-linking of this polysaccharide long chains, leading to the formation of the peptidoglycan cell wall. However, E. coli is a gram-negative bacterial cell. Staphylococcus aureus is a gram-positive bacterial cell. So, one difference between the gram-negative bacterial cell and the gram-positive bacterial cell is that the gram-positive bacterial cells have much thicker peptidoglycan layer. The gram-negatives have much thinner peptidoglycan layer. But also in the composition of the amino acid residues that are present in the short polypeptide chains that come out of these polysaccharide chains, the amino acid composition is different as well as the nature of cross-linking, right? The nature of cross-linking of these um, this adjacent polysaccharide chains is different. In case of the gram-positive bacteria like Staphylococcus aureus, the adjacent polysaccharide chains are linked by a pentaglycine bridge, right? Whereas in case of this gram-negative peptidoglycan chain, the the polypeptide chains arising from this adjacent polysaccharide chains made of N-acetyl glucosamine and N-acetyl muramic acid are joined directly by formation of a peptide bond. So the cross linkages are very different between gram-negative bacteria and gram-positive bacteria, which is also of some importance considering human health and disease. Like several of these antibiotics can specifically inhibit only gram-positive bacteria, but not gram-negative bacteria because of these different cross linkages and different composition to certain extent of these amino acid residues that are present in the polypeptide, these short polypeptide chains that are coming in, because different enzymes are necessary and are at play in case of gram-negative versus gram-positive organism. So that also gives you this handle to selectively inhibit gram-negative bacteria or gram-positive bacteria. Right? Okay. So this slide also gives you a three-dimensional idea of the organization of the cell envelope of gram-negative bacteria and gram-positive bacteria. So on the left-hand side, you have the gram-positive cell envelope, which consists of the lipid bilayer of the cell membrane or the plasma membrane. And on top of that, you have a very thick peptidoglycan layer, again, consisting of this long polysaccharide chains that are formed as a result of um, polymerization of N-acetyl glutamic acid and N-acetyl glucosamine and N-acetyl muramic acid. And then these polysaccharide chains are again cross-linked because of cross-linking of the um, polypeptide chains that are coming out of the polysaccharide chains. And also you have teichoic acid as a major component of this gram-positive peptidoglycan cell wall. But you can also, so this provides mechanical strength. It's a tough layer that is present outside the plasma membrane. It provides mechanical strength to the plasma membrane, but also it is porous, right? That allows nutrient molecules or other signaling molecules, molecules which might be important for bacterial quorum sensing to enter from this extracellular space to this porous peptidoglycan layer into the bacterial cell by passing through the plasma membrane. Gram-negative cell envelope, on the other hand, if you can see, then it consists of the cell membrane, right? Then a very short, then this is the periplasmic space. There is a gap between the peptidoglycan and actually this gap between this peptidoglycan outer membrane and this, I mean, the gap between the outer membrane and the plasma membrane, where lies a very thin peptidoglycan layer, which again provides mechanical support as well as protection against osmotic stress. So, and then here also the peptidoglycan, there's a composition of these amino acid residues will be a bit different as compared to gram-positive bacterial peptidoglycan, and the cross linkages will also be different, and the enzymes at play will be different. The outer membrane is also is outermost layer, and it consists of it also consists of a lipid bilayer, just like the plasma membrane, but it is quite unique in the lipid composition. It has several lipids that are very unique to the outer membrane and are not present in the bacterial cell membrane or the plasma membrane. And this outer membrane also has, the outer membrane is a lipid membrane. So that has specific proteins like these porine proteins, which specifically allow several molecules to get into this, to get into the periplasmic space via crossing this outer membrane and eventually get into the bacterial cell by passing through the respective proteins that are present in the bacterial cell membrane or the plasma membrane. Yeah, so the difference in the architecture or organization of the cell wall and cell envelope of the bacterial cells was actually discovered later. Initially, the first hallmark experiment that showed that there are two different classes of bacteria for some reason was a staining protocol developed by Christian Gram. So how in this Gram staining protocol, it was found that there seems to be two different classes of bacteria which respond differently to the gram staining protocol and to the gram stain, which actually shows different response in terms of retaining the gram stain and taking up the counter stain. So let me tell you the gram staining protocol in brief. In gram staining protocol, primarily the smear of the bacterial cells are stained with the gram stain, which is the primary stain, that is crystal violet, right? So these crystal violet molecules are trapped by the cell envelope in case of both gram positive and gram negative bacterial cells. Now, basically, if to begin with, for example, let us not, I mean, we do, for example, to begin with, Christian Graham had not expected that there might be two different types of bacterial cells. So he initially saw that after staining, all the bacterial cells took the color. Took the color because of adsorption of crystal violet stain to all of the bacterial cells. Then this iodine was added as a mordant, which further stabilizes, which makes a complex with this crystal violet, and so that this crystal violet iodine complex is very nicely retained and uptaken by these bacterial cells. And the cells, all of the cells remain purple because of uptake or adsorption of this crystal violet iodine complex. Then, in Gram's staining protocol, a decolorizer is added. A destaining to check whether the cells are losing the stain molecules or not. There, what happens is that what happens is that some of these bacterial cells, so this decolorizer was alcohol. Okay, so alcohol was added as a decolorizer to remove perhaps this excess. In his initial protocol, he may have added this decolorizer just in the hope that this excess crystal violet iodine stain will be removed, and he will be able to see the bacterial cells very nicely. Only the bacterial cells which are retaining the. He hoped that maybe all of these bacterial cells will be retaining the stain, and he just staining or removing the excess background stain will help him see the bacterial cells nicely. But what happened is that some of the bacterial cells in this step failed to retain the crystal violet iodine complex, while some of the bacterial cells still retained, which he hoped will happen. Some of the bacteria are able to retain this crystal violet iodine stain, whereas some of the bacterial cells were not able to. And upon staining with another stain, which is safranin or counter stain, to visualize the bacterial cells which have lost the crystal violet staining. So these bacterial cells which lost the crystal violet staining now take up the counter stain, which is safranin, whereas the other bacterial cells which are already retaining the crystal violet stain, they don't. They still stay purple. So this was the first. This gram staining protocol was the first experiment which showed that bacterial cells are of two different types, depending on their response to this entire gram staining protocol. That somewhere, even after addition of this decolorizer, we're able to retain the gram stain, that is this crystal violet iodine complex, whereas some cells were destained. They failed to retain the crystal violet iodine complex, and they took up the counter stain, which is safranin, and they appeared pink instead of appearing violet. And later it was found that this differential response to the gram staining protocol was actually because of the different composition and different architecture or organizational status of the bacterial cell envelopes. So actually what happens is that these gram-negative bacterial cells have the outer membrane as the outermost layer. So they initially trap the crystal violet iodine complex on the outer membrane. When you add alcohol, the alcohol washes away or wipes away this outer membrane lipid bilayer considerably, and with that the crystal violet iodine complex is also washed away. That's why they lose the purple color of this gram stain. And when you next stain them with safranin, they take up the counter stain and they appear pink. Whereas the gram-positive organisms, which do not have the outer membrane, they actually capture the crystal violet iodine complex in their thick peptidoglycan layer. And upon addition of alcohol, there is no outer membrane that is washed away. Instead, what happens is that it causes dehydration of this peptidoglycan layer, which makes the pores to shrink even more. That means this crystal violet iodine complex, which was trapped within this porous peptidoglycan, even gets much nicely trapped within the peptidoglycan shrinked, dehydrated peptidoglycan layer, and they retain the crystallization and even don't take up. Okay, so yeah, so now let's look into the cell wall that is present in the eukaryotic plant cells. So the animal cells are also eukaryotic cells, but they don't have a cell wall. Whereas the plant cells, um, the plant eukaryotic cells, they have a cell wall. Because the plant cell wall is also made up of these polysaccharides, primarily cellulose, hemicellulose, and pectin. Primarily cellulose, which is embedded in a matrix that consists of pectin and hemicellulose, and various other glycoproteins and enzymes are also there, which play important role in the synthesis of this cell wall, as well as the remodeling of the cell wall, depending on the different status of the plant cells and plant metabolism, as well as environmental cues or signals. So the plant cells also have this cell membrane or the plasma membrane. Now these young plant cells, the growing plant cells, very actively photosynthesizing and metabolizing plant cells, so they do not have a very thick cell wall. They have just this middle lamella, which primarily consists of pectin as kind of extracellular matrix which holds together, which kind of gels together or keeps together the plant cells to form tissues out of this large number of plant cells. But in those tissues of the plant which need to provide, which are not so actively metabolizing, not so growing actively, and instead need to provide the mechanical strength or mechanical support or the rigidity for allowing the plant cells to, for example, grow so tall against gravity to compensate for their inability to move. They grow taller and taller in search of more and more resources like sunshine, sunlight for increasing their photosynthesis. So some of these specialized tissues, they actually generate much increased levels of the cell wall um, with time. So beneath these initial middle lamella, which primarily consists of this extracellular matrix polysaccharide pectin, So yeah, as we were discussing that in those tissues which are primarily needed to provide the mechanical strength and support the plant cells for them to grow tall and in search for resources, especially sunlight for photosynthesizing. There what happens is that beneath the middle lamella is deposited the first layer of this polysaccharide containing cell wall matrix materials like cellulose, hemicellulose, and other lignin or cutin to form the primary cell wall. And then extra layers of even like extra more layers of cell walls are being deposited gradually with time and as the plant tissues age, which are called the secondary cell wall layers, which lead to even thickening of the cell wall. And because to serve the purpose or serve the need of providing the extra mechanical support as a plant grows taller and taller, uh, mechanical support to the lower parts of the trunk of the tree as the tree grows taller and taller, or the branch grows thicker and thicker and longer, right? Okay, so but consider this fact that just like we mentioned initially, that the cell wall, the plant cell wall is also porous, and it allows the movement of nutrient molecules and important signaling messenger molecules between the plant cells for allowing cell-to-cell communication and cell-to-cell crosstalk, which is necessary for organization of the cells and functioning of the cells as a group, like tissues in different parts of the plant. As well as it allows the formation of the cell-to-cell junctions, which are called the plasmodesmata, which are very important for the plant cell. So on the whole, each cell's cell wall allows the neighboring cells to interact with one another and form uh, these tissues, and allow these groups of large number of cells to function as a single group or tissue of different plant parts. Yeah, this figure again shows you the organization of the polysaccharide materials of the plant cell wall in a bit more details. Basically, the primary component is the polysaccharide cellulose, which is being secreted by the plant cells themselves. They are basically secreted outside the plant cell, and the cellulose forms cellulose fibrils, which get embedded in the matrix of hemicellulose and pectin to form the cellulose microfibrils. Actually, get microfibrils or microfibers get embedded in the matrix of pectin and hemicellulose to form the larger fibrils, which accumulate beneath the middle lamella to form the primary cell wall, and then the additional cell wall layers that are being added forms the secondary cell wall. And in many cases, there are these special components like lignins, which provide even additional rigidity and mechanical strength and reduces the fibrosity, to be honest, makes it less fibrous and more rigid, much less flexible in some specialized tissues of the plant cells, especially the wood of the tree, for example. Okay, so after we have seen the outermost layer of these cells, like the bacterial cells, as well as the plant cells, which was the cell wall. Let's come to the layer that lies within the cell wall, but is very important even for the existence of a cell, which is the plasma membrane. Plasma membrane is the boundary of the cell, which really makes the cell a cell. It's the without the plasma membrane, there is no existence of the cell only, and plasma membrane ensures the maintenance of a homeostasis or constancy of a particular environment, a very selective and regulated manner within the cells, despite changes in the extracellular environment. Right? So it allows the maintenance of the cellular homeostasis. Now the plasma membrane is primarily a lipid bilayer. Right? It's a lipid bilayer within which the proteins, several different protein molecules are embedded. And this, as we will see in the next slide, the fluid mosaic model of the plasma membrane, that these protein molecules are embedded and they are, floating in this sea of lipid. But this lipid bilayer is not a static one. It's a very dynamic lipid bilayer where all of these molecules are continuously moving on and diffusing within this lipid bilayer within a single leaflet of the lipid bilayer, as well as there are also exchanges between the two different leaflets of the lipid bilayer. And also these this lipid bilayer of the plasma membrane is not very homogeneous. It's not like that that all the proteins are equally and homogeneously distributed all over the lipid membrane, this plasma membrane, or it is also not the case that all the lipids are homogeneously and uniformly distributed all through the plasma membrane. So neither the lipids nor the proteins are homogeneously or uniformly distributed all across the plasma membrane. That means there lies some heterogeneity in the organization of the lipids and proteins. So there are some clusters which have unique lipid compositions, like they are much more enriched in certain lipids like sphingolipid, uh, cholesterol, and certain forms of proteins like GPI anchored proteins, which play unique functions because of being present in these specialized regions which are called the lipid. In many cases, they are important signaling hubs of the proteins and various other. Okay, so as the fluid mosaic model says, that you can appreciate that the plasma membrane is a lipid bilayer, right? It consists of two leaflets of lipid molecules, and you have the proteins which are floating in this sea of lipid. Everything is very dynamic. All the molecules are constantly in a state of movement and diffusion within this within each of these leaflet of the lipid bilayer of the plasma membrane. There are several different types of proteins as well, which depending on the nature of their incorporation in the lipid bilayer, there are transmembrane proteins or there are proteins that are just associated with a single leaflet of the um, lipid bilayer, and these proteins perform different a variety or a range of diverse cellular functions. But one important component is that this is the extracellular part of the cell, and this is the cytosol. So the lipid, the plasma membrane, which is primarily made up of this lipid which is primarily a lipid bilayer, needs some mechanical support and rigidity uh, to ensure that there is a particular cell shape, and that is actually provided by the cytoskeletal elements, right? Which also we will see in some detail in the next slides. So there is a base of cytoskeletal elements, primarily in this case, the actin cytoskeletal elements, actin microfilament cytoskeletal elements, which provide the mechanical strength and the base for beneath or towards the cytoplasmic phase of the plasma membrane, which is made up of this, which is a lipid bilayer, which is made up of so many different types of lipids like phospholipids, glycerolipids, cholesterol, sphingolipids, etc. Yeah. So the plasma membrane lipid bilayer is actually made up of amphipathic lipids, right? What that means is that the lipids with which the plasma membrane is made up of consists of polar hydrophilic head groups and hydrophobic, um, that is non-polar hydrocarbon chains, right? Hydrocarbon fatty acid chains, or hydrocarbon fatty acid tails. So, which we have already discussed in one of our previous classes, where we looked at the different kinds of lipids. So, since phospholipids are the major kinds of lipids, along with also glycerolipids, which consist of glycerol, two fatty acid chains attached to glycerol, and then the phosphate group attached. And so, this phosphate group forms your hydrophilic polar head group, whereas the hydrocarbon chains, the long hydrocarbon chains forms the non-polar or hydrophobic tails. Right? Now, when these amphipathic lipids are present in this aqueous environment, that is, they are present in water as a solvent. There, what happens is that these hydrocarbon chains, which are non-polar and are not able to form hydrogen bonds with water, and they're not able to interact with water, they hate water, right? and water hate this hydrocarbon chains. So that is energetically unfavorable. So as you go on increasing the concentration of these lipids, what happens is they self-assemble, right? They self-assemble in such a manner that these hydrocarbon chains try to hide themselves away from water. Whereas this hydrophilic uh, head molecules, this which primarily consist of this phosphate residues, are capable of forming hydrogen bonds with water, and so they can be exposed to the water or the aqueous environment. So they self-assemble themselves in the form of a lipid bilayer like this, which is shown here, which is around 4 nanometer thick, where you can see that these hydrophobic hydrocarbon chains are hidden in the inside. They're packed and hidden inside the lipid bilayer, whereas these hydrophilic polar head groups are present exposed to the aqueous environment. Right? And there can be different lipids, phospholipids, sphingolipids, cholesterols, which make up the plasma membrane. Now, after formation of, so you can think that now there is one more level of complexity or self-assembly actually. That so as you can imagine, as we have already discussed, that with increasing concentration of these phospholipids or glycerolipids, basically amphipathic lipids, you see the lipid self-assemble in the form of a of this bilayer. Now see this thing that it's a planar bilayer, right? It's a planar like a sheet, but with this planar lipid bilayer consisting of these amphipathic lipids is still energetically unfavorable because at the edges, these hydrophobic residues, right, these hydrophobic hydrocarbon chains of these phospholipids or these amphipathic lipids basically are still seeing water, right? So that is energetically unfavorable. So they try to even hide themselves even more. And what happens in that case is that this planar phospholipid bilayer, which has these edges that are exposed to water, closes upon itself, right? It folds and closes upon itself to form a completely sealed spherical phospholipid bilayer, right? Which is now energetically favorable because the hydrophobic hydrocarbon chains will now be completely hidden from the water, and there will be these hydrophilic head groups which will be facing the water both inside the lipid compartment and outside the lipid compartment. So this gives you the idea how these amphipathic lipids can self-assemble to form this planar bilayer in the beginning, which eventually closes on itself spontaneously to form a completely sealed compartment that is formed of this lipid bilayer, which is reminiscent of the first cells that would have arose. That is the protocells, right? And still, in case of these cells, this is the thermodynamic driving force that makes these amphipathic lipids assemble to form the lipid bilayer that is present in the cell. Okay. So any question till now? Okay. So let's move on. Now, as we've already mentioned in the fluid mosaic model, that there is a considerable fluidity and dynamics in the lipid bilayer, right? Where these lipid molecules are continuously diffusing and moving around, and there are also these lipid rafts which are also moving around in some manner, some regulated manner. And the proteins are also moving within the lipid bilayer, and the proteins are also undergoing various conformational changes for performing their functions. So there is a fluidity of the lipid bilayer. Now, the fluidity of the lipid bilayer depends enormously on the composition, especially the lipid composition of the plasma membrane, of the leaflets of the plasma membrane. This we had also discussed while discussing about the different types of amphipathic lipids like phospholipids, glycerolipids, cholesterol. That, for example, even in the case of these phospholipids, if we have saturated hydrocarbon chains versus unsaturated hydrocarbon chains, and saturated hydrocarbon chains, since they do not have this kink inducing double bond, since they do not have the kink forming double bond, they can stack very nicely, right? So they decrease the fluidity and increase the packing, or perhaps the strength of the membrane. Whereas these unsaturated hydrocarbon chains or the phospholipid molecules, because of having this double bond, which introduces a kink in the molecule, right? Hello, any question? Okay. So this unsaturated hydrocarbon chains of the phospholipids, which have a double bond, and because of that, there is a kink which is introduced. So they reduce the fluidity of the plasma membrane. Yeah. They did, sorry, because of these unsaturated lipids, unsaturated hydrocarbon chains of the phospholipids, because of having this double bond which introduces a kink, so they cannot pack very nicely, right? They cannot pack very nicely, so they increase the fluidity. So saturated lipids or saturated hydrocarbon chains, as they pack very nicely, they reduce the fluidity and increase the stability or the strength of the membrane. Whereas the unsaturated lipids, which have this kink because of the double bond, the carbon-carbon double bond, are not able to pack very nicely, and they increase the fluidity, whereas reduce the strength or rigidity of the membrane. Cholesterol, on the other hand, is cholesterols and sphingolipids. They increase the rigidity, right? They increase the rigidity and the strength of the membrane, but reduces and stiffens the bilayer and reduces the fluidity. And because of that also, increased levels of cholesterol allows the packing of lipid molecules even within this lipid bilayer to form a bit different uh, sub-phase of the lipid bilayer, which are called the lipid rafts, which float like islands within this sea of lipids, right? So, in short, the membrane fluidity is highly regulated, and that is regulated by regulating the composition of the lipids that are present in each leaflet of the lipid membrane bilayer of the plasma membrane. Now, there are different types of proteins. The proteins that are present in the plasma membrane can be classified into several different categories depending on their nature of incorporation within the plasma membrane. Right. So some of these proteins, so many of these proteins come under the category which are called the transmembrane or integral membrane proteins, right? So these integral or transmembrane proteins have one. They span the entire lipid bilayer. They span both the leaflets of the lipid bilayer, right? Now they can have one section. I mean, they can have just a single section of the protein. For example, this alpha helices, this alpha helix, which has hydrophobic amino acid residues and therefore can be incorporated within this lipid bilayer, or it can have several sections of the protein that span the um, plasma membrane. So that this will be called the first one. The is called the single pass transmembrane protein, since it has a single secondary structural element or of the protein that is incorporated within the plasma membrane. And the second one, these others will be called the multi-pass transmembrane protein or integral membrane proteins. Many of these transmembrane proteins or integral membrane proteins act as important transporters, right? Transporters means these proteins ensure the specific are important for the selective permeability of the cells. They ensure specifically certain molecules to come inside the cell from the extracellular space or go outside the cell that is from the cytoplasm to the extracellular space. There are also other proteins like membrane-associated proteins. So in these membrane-associated proteins, they are not transmembrane proteins. These proteins are not completely incorporated within the two leaflets, and they're not spanning the entire lipid bilayer. Instead, they're present in any single leaflet of the lipid bilayer, right? they can be important proteins which like they can be important scaffold proteins which allow other proteins to get recruited to the membrane for doing certain function. And there can be these lipid linked proteins, right? Lipid anchored proteins, or in other words, which can be a major class of which are GPI anchored proteins, many of which being important signaling proteins as well as important for other cellular functions like endocytosis and etc. So in the instance of these GPI anchored proteins, what happens is that these proteins, the proteins are associated with a single liplet of the lipid bilayer, not because of incorporation of any protein secondary structure or proteins section or fragment, but because of anchoring of a lipid molecule, right? Because of anchoring of a lipid molecule, which is covalently attached to this protein into one leaflet of the lipid bilayer, right? So these proteins, which are lipid-linked proteins, for example, glycosyl phosphatidylinositol or GPI anchored proteins, here the proteins are post-translationally modified with a lipid anchor like GPI, glycosyl phosphatidylinositol, which are covalently linked to the protein, and this GPI anchor is actually associated. This lipid is actually anchored within one leaflet of the lipid bilayer. That's why these proteins are also attached to the one leaflet of the plasma membrane. And there are several other proteins which I was already mentioning in this case as well. Like there can be these membrane associated proteins or transmembrane proteins, which which can have several interacting proteins, right? There can be several interacting proteins present in the cytosol or the extracellular matrix, which can interact with some of these membrane transmembrane proteins or these membrane associated proteins, and thereby get recruited to the plasma membrane and do certain important functions, right? So those are also a category of proteins which are found to be quite associated, but dynamically with the plasma membrane. So they are much more dynamic as compared to these transmembrane proteins or membrane associated proteins, but they also have this capability to kind of get recruited to the membrane because of interacting with these transmembrane proteins or the membrane associated proteins. They are already present at the plasma membrane. Okay, so and as you can already imagine that there are so many different proteins that are present in the plasma membrane or the cell membrane, and they perform a wide range of diverse cellular functions, right? They can be transporters, which are essential for maintaining the selective permeability of the cells. There can be ion transporters, there can be transporters of nutrients like glucose, um, ensuring the movement of molecules in a very selective manner in both the directions, that is from the extracellular space to the cytosol, as well as from the cytosol or cytoplasm to the extracellular space, right? An important category of proteins are adhesion proteins or anchors, which ensure the adhesion or anchoring of the cells to the extracellular matrix, as well as cell-to-cell adhesion, for ensuring that that a large number of cells are anchored and adhered to form and function like a group of cells, that is form tissues and function like tissues, right? A major category of plasma membrane proteins are receptors, which are very important functions in terms of signal transduction. That is, these proteins bind one particular molecule, which is called a ligand, in the extracellular space. And when this ligand molecule, which is present in the extracellular space, because of binding this ligand, which is present in the extracellular space, it can recognize a specific signal or stimuli that is present in the extracellular space. And after recognizing that, what will happen is there will be conformational change of the protein on the cytosolic phase of the plasma membrane. And because of that, there will be relay of the signal. There will be relay of the signal in the cytosol because of selective recruitment of several different downstream protein molecules that are present in the cytosol to this cytosolic phase of this receptor molecule. So essentially, many of these plasma membrane proteins, mostly transmembrane plasma membrane proteins, which function as receptors, so they're capable of recognizing and specifically binding one particular signal or stimuli that is present in the extracellular space and in response to that, show conformational changes in the cytosolic domains of this particular protein, recruit specific downstream proteins from the cytosol, and relay the signal and generate a certain signal in the cytosolic end of the protein and relay the signal via a set of proteins and eventually lead to a cellular response in to the extracellular signal or stimuli, right? And obviously, definitely, last but never the least, large set of membrane-associated proteins have important enzymatic activities. Like for example, kinases, which phosphorylate proteins. Many of these kinases, which are partner molecules or interacting proteins of many of these receptors, play important role in signal transduction as well as various other processes. As well as many of these enzymes will also be those proteins which regulate the lipid composition of the plasma membrane, which is very essential for several functions, like maintaining the membrane fluidity, as well as in several different aspects like cell survival and cell death or apoptosis, where there comes an interesting set of proteins called flipases, which move proteins from the inner leaflet or the cytosolic leaflet of the plasma membrane to they move phosphatidylserine from the inner leaflet of the um, plasma membrane to the extracellular leaflet of the plasma membrane. So phosphatidylserine is typically not present in the extracellular leaflet of the plasma membrane. Only when the cell is under stress and to signal apoptosis or programmed cell death, the flipases come into picture and they transfer phosphatidylserine from the inner leaflet of the plasma membrane to the outer leaflet of the plasma membrane. Okay. So now come to the next part of this week's content, which is also a very important component of the eukaryotic cells as well as the prokaryotic cells, but more elaborate and complex in the eukaryotic cells. So primarily cytoskeletal elements and cytoskeletal architecture and organization of the eukaryotic cells was discussed. Um, so this plasma membrane is actually a very wobbly and floppy structure as expected because it's made up of just a lipid bilayer, right? So it should not, it's very difficult for that to have a mechanical strength or rigidity to ensure a certain shape or structure, right? So this is it's a very nice analogy that, for example, to make one tent, you have primarily two components. One is your rods, and the other one is a tent cloth that you will eventually spread over the architecture or the bones of the rods to have your tent. So if the rods are being removed, then the tent cloth cannot itself assume the shape of the tent. It will all collapse, right? So this mechanical strength or support that is provided by these rods in case of a tent is necessary for allowing um, the tent cloth to take a certain shape and perform so many different functions as a tent. Just like that, in case of the cells as well, um, beneath the beneath the plasma membrane, which is made up of this lipid bilayer, lies a comprehensive network of cytoskeletal elements beneath the plasma membrane as well as all through the cytosol or the cytoplasm of the cells, which is very important for allowing the cells to assume a certain shape and stabilize the cells, have a certain structure, have a certain organization, or have a certain organization, even in terms of the spatial organization of different subcellular compartments, like cellular organelles. For example, within the cell, as we have already seen, that there is a particular spatial distribution of different organelles. For example, the nucleus is present at the center, and then there is the endoplasmic reticulum, the Golgi complex, the mitochondria, lysosomes, and however, this organization is also not fixed. The organization also keeps on changing depending on different cell cycle stages and cellular stress or cellular metabolism, extracellular signals, stimuli that comes in. So in that as well, for the spatial for regulating the spatial distribution of these subcellular compartments, membrane-bound subcellular compartments and organelles. Also, this network of cytoskeleton provides a very important um, base or mechanical support structure to which the organelles of the subcellular compartments can be anchored, and thereby their subcellular localization can be regulated, right? So the cytoskeleton is a very important component in providing mechanical strength, support for the cellular shape, as well as spatial organization of the subcellular compartments or organelles within the cells by anchoring those organelles to certain cytoskeletal elements, as well as the cytoskeletal elements act as major transport highways for the cell. Many of the vesicles are carried by proteins called motor proteins, which run on these microtubules for transporting these vesicles from the nucleus to the plasma membrane or plasma membrane to the nucleus. It is in both directions, right? Um, Over long distances, if you consider in case of cells, for example, even in case of axons of the neurons, which are pretty long, there are also these extensive microtubular networks allow the transport of these vesicles on several different organelles, having important cellular cargo um, from uh, one uh, from one place of the cell to the other place of the cell. These cytoskeletal elements, however, are again not static structures. They are very dynamic. They're very dynamic in this sense that the directionality changes. They're made as well as they're broken. They're assembled as well as disassembled in a certain manner to achieve a certain goal, right? Depending on the cellular goals like cellular migration, cellular mobility, cellular um, movements of the cellular plasma membrane, for example, in case of endocytosis or phagocytosis, etc. So the cytoskeleton also, cytoskeletal elements are very dynamic, and the dynamism is very actively regulated because of the balance between the assembly of the cytoskeletal of the monomeric units of the cytoskeleton as and the disassembly by regulating the because of the balance the regulation of the assembly and disassembly of the cytoskeletal elements, right? So that makes the cytoskeleton quite dynamic. So this is exactly what I was mm, trying to mention in the previous slide as well, that the cytoskeletal elements are quite dynamic. Now, the cytoskeletal elements, be it microtubules, actin filaments, or intermediate filaments, are made up of proteins, right? They are made up of proteins, whereas the lipid membrane, whereas this plasma membrane or the cell membrane, or even the membrane of the subcellular compartments or organelles are made up of lipids. So the membranes are made up of lipids, the plasma membrane as well as the membrane of the subcellular compartments and the organelles made up of lipids. They're lipid bilayers made up of amphipathic lipids, whereas the cytoskeletal elements are formed as a result of assembly and polymerization of protein subunits, right? And so there are these small soluble subunits, small soluble protein subunits, which polymerize to form large filamentous polymers of cytoskeletal elements, be it microtubules, be it actin filaments, microfilaments, or intermediate filaments. But they're very dynamic, and their assembly disassembly balance, right? Their assembly disassembly balance is regulated by various factors: cellular metabolism, extracellular signal or stimulus, as well as other cues. So, so you can see this is a very nice example. That initially, cell was trying to move, and the cell was actually trying to move and migrate in this direction, right? Maybe because at that point of time that there was a nutrient gradient in this direction. That is, higher nutrient on the left and lower nutrient on the right. Lower nutrient concentration on the right, higher nutrient concentration on the left. So the cell was trying to grow in that direction or move in that direction. But later, because of some reason, when the nutrient gradient has changed, for example, it has changed in this direction now. That now the high concentration is on the right end and the low concentration is on the left end. Or for example, take this example that this is a macrophage, right? This is a macrophage with a phagocytic cell which is chasing a bacteria. okay, or it's a neutrophil which is chasing a bacteria that has invaded your body. Then for initially the bacteria was moving in this direction, for example. So the cell was also trying to move in this direction, but now the bacteria is moving in this direction. So the cell has to reorient itself, right? It has to reaccess the situation. It has to reorient, and now it has to move towards the right. So this is possible because initially the cell was able to move and migrate towards the left direction, because all of these the cell membrane's movement and changes, be it protrusion or invagination, is governed considerably by the remodeling of the cytoskeletal elements that are present beneath the plasma membrane. So this cell was able to move in a certain towards the left because the underlying actin cytoskeleton and various other microtubular elements were getting polymerized in this direction, such that the cell membrane also protrudes and elongates in this direction, enabling the cell to move from the left towards from the right towards the left. However, when the bacterial movement changed, so the ligand concentration must have also changed, or the bacterial or this eukaryotic cell, this macrophage or neutrophil has sensed that okay, it now needs to move in a certain different direction. So therefore that now it needs to move in the complete opposite direction. So it needs the actin polymerization in a complete different end. It now needs it in this end, the right end. So there that is being done, that is made possible because of the uh, this capability of assembly as well as disassembly of these units or monomeric subunits of this actin of these cytoskeletal elements. So there will be first disassembly of these subunits from these filamentous cytoskeletal elements, and then again these subunits will be specifically polymerized and elongated and assembled on this right end, so that the cell now is able to elongate its membrane and form its protrusions towards this right end and chase the bacteria, right? And eventually phagocytose it. And even in this case of phagocytosis, there will be membrane invagination, which will ensure the cell to engulf one bacteria, for example, and eventually it will engulf the bacteria. So that will also need behind this membrane remodeling. Always remember that there will be remodeling. There needs to be remodeling of the underlying cytoskeletal elements to ensure one particular membrane event, be it phagocytosis, cellular movement, that is cell motility, cell migration, or endocytosis for physiological cellular events, etc. Uptake of nutrients, for example, etc. Okay, so let's look in a bit more details about the different cytoskeletal elements and what are these cytoskeletal elements made up of. So the eukaryotic cells have three different cytoskeletal elements: microtubules, microfilaments, and intermediate filaments. Right now, each of these cytoskeletal elements have some unique functions. Okay, have some unique functions of their own, whereas they also have several functions which are quite shared. Like there is some redundancy. That is, there are some functions which any of these cytoskeletal elements are capable of executing. So there are some shared functions or redundant functions, and there are some unique functions which only a particular type of cytoskeletal element is capable of doing or executing. Microtubules are actually microtubules are hollow tubular cytoskeletal elements, which are hollow tubular long cytoskeletal elements, unbranched cytoskeletal elements that majorly serve as the highways, the transport highways of the cells, act as important cytoskeletal elements in terms of anchoring of the subcellular compartments and subcellular organelles, as well as transport and traffic of the organelles. So they are the transport highways of vesicles having cellular cargo, transport highways for the movement of organelles from one part of the cell to other part of the cell. In case of neurons, for example, take this example. In case of neurons, the movement of these vesicles or transport of these vesicles that are filled with neurotransmitters from the cell body or the soma of the neuron to the end of the axon for the delivery of the neurotransmitter, eventually to the synaptic cleft for the transmission of a signal, right? So these microtubules, which are hollow tubular unbranched long structures, they act as major highways for the transport of these vesicles and organelles, for the anchorage of organelles. Whereas these microfilaments, these actin microfilaments, they are fibril-like. They're fibrillar assemblies of actin subunits, and they primarily are involved in ensuring the cell shape, cell migration, and remodeling of the plasma membrane, like invagination of the plasma membrane or extending the protrusions of the plasma membrane. For example, in case of phagocytosis or endocytosis. So they are involved in maintaining the cell shape, cell-cell junctions, for example, cell-cell adhesions, cell extracellular matrix adhesions, right? Cell-cell adhesions, cell extracellular matrix adhesions, cell motility, cell migration, as well as remodeling of the cellular membrane for processes like endocytosis and phagocytosis. Right? The intermediate filaments, so basically the actin filaments are present mostly. The actin filaments are present mostly just beneath the plasma membrane, and therefore they are capable of executing these functions like cell shape, cell motility, cell migration, and membrane remodeling, like membrane invagination or phagocytosis, endocytosis, etc. Protrusions during cell migration and cell mobility. Whereas microtubules, they mostly, as we will see, they mostly start to get formed near the nucleus, like by the side of the nucleus. So from the center of the cell, almost, they start forming and then they migrate to the periphery of the cell, and therefore they are majorly these highways for transport, for bidirectional transport, both from the nucleus towards the plasma membrane, as well as from the plasma membrane towards the nucleus, right? And as well as they act as important anchorage elements for maintaining a particular spatial distribution of the subcellular organelles. Intermediate filaments are distributed, I mean, almost all over the cell, but also they have certain, I mean, they can be of several different types depending on which protein subunit they're made up of and the subcellular compartment in which they're localized. For example, a good example, a very common and important example of these intermediate filaments are the nuclear lamina, the cytoskeletal elements, these intermediate filaments, which are made up of lamine proteins that are present just beneath the nucleus, the nuclear membrane, towards the nucleoplasm, are important for providing mechanical strength to the nuclear membrane, right? For maintaining the integrity of the nucleus. So basically, this table provides you a nice summary and comparative view of these three different cytoskeletal elements of the eukaryotic cell. So as I already mentioned, that the microtubules are made up of tubulin proteins. So there is alpha tubulin and there is beta tubulin, which first forms heterodimers. And then these heterodimers assemble in the form of protofilaments. And then these protofilaments assemble in a certain manner to form a helix. Actually, they assemble these protofilaments, the chains of tubulin dimers, assemble in a spiral manner with relative to one another, leading to the formation of a tubular, right? Leading to the formation of a tubular or hollow fibrillar assembly, hollow, not fibrillar, hollow tubular assembly, which will be called the microtubules. That act as the major highways of the cells, because the motor proteins, kinase and adenine, are capable of getting recruited on these microtubules, and they transport cargoes in a particular direction. And one very important and common example or function of these tubulin is in the form of spindle fibers, right? During cell division, during the mitosis and meiosis, cell division in the anaphase, like in the metaphase, all the chromosomes, all these chromatin is condensed in the form of chromosomes, and to the centromere, to this kinetochore, is attached the spindle fibers. And during the anaphase, there is shortening of these microtubules of the spindle fibers. Because of that, spindle fibers are made up of microtubules. cytoskeletal elements, and because of shortening of these microtubules or the spindle fibers, there is movement of the segregation of the sister chromatids to the two different ends, and eventually into two different daughter cells. Actin filaments, on the other hand, are they are again they are made up of actin subunits, are made up of actin protein subunits, which polymerize to form protofilaments. Now two actin protofilaments are intertwined or intertwisted, right? Intertwined or intertwisted into a right-handed helix-like fibrillar assembly, and they are present primarily just beneath the plasma membrane, and are important in providing strength and mechanical rigidity to the plasma membrane, as well as governing the shape of the cell and the plasma membrane, as well as governing this invagination, protrusion, cell migration, cell movement, right? As well as important in the cell division, like during the cytokinesis. And the intermediate filaments, which are distributed all throughout the cells, and they can be of several different types again, depending on the protein component, which has polymerized to form this intermediate filament, and depending on the subcellular compartment in which it is located. These actin filaments, however, it is worth mentioning that in tissues like the muscles, the contractility, the force that is generated, this expansion or contraction of the muscle, right? The expansion or contraction of the muscle is because of the interplay of actin and myosin cytoskeletal elements, right? It's because of the interplay of actin and myosin microfilament cytoskeletal elements, which generates the force because of contraction or relaxation, that is expansion of the muscle fibers. So let's just look in a little bit more details about the microtubule cytoskeletal elements and the actin microfilament cytoskeletal elements. So as we have already mentioned that these micro the microtubules are made up of tubulin proteins, right? Tubulin protein subunits. So they consist, and so the tubulin protein subunits are of two different types: alpha tubulin and beta tubulin. Now, alpha tubulin and beta tubulin first heterodimerizes. One minute. Yeah, so this alpha tubulin and beta tubulin protein subunits, they first heterodimerize in a GTP. They bind this subunits actually bind guanosine triphosphate as a nucleotide, and that dimerization and polymerization is dependent on this nucleotide binding, right? That is GTP binding. Now, what happens is that this alpha tubulin binds when this one GTP molecule is present at the interface between this alpha tubulin and beta tubulin. Beta tubulin, on the other hand, can be actually bound to GTP or GDP. So that is interchangeable. So there can be so GTP bound beta tubulin makes this heterodimer much stable, right? Makes this heterodimer much stable and facilitates polymerization and extension of the microtubule. Whereas hydrolysis of this GTP of beta tubulin to GDP, that is diphosphate, makes this heterodimer less stable and promotes disassembly of the microtubule. So what happens initially, let's see the polymerization or assembly of heterodimers of alpha tubulin and beta tubulin first. So first you have the tubulin heterodimer, which is your most functional subunit. Then these heterodimers join head to tail, in a head to tail manner, right? This is one dimer, this is one dimer, this is one dimer. These dimers join with one another to form these single filaments, which are called the protofilaments, right? And then several of these protofilaments, to be precise, 13 of these protofilaments will assemble, right? Will assemble laterally, right? That is one after the other in such a manner that it will in a spiral manner. Okay, in a spiral manner they will assemble. Okay, to form a hollow tubular microtubule filament. Okay, now in this assembly there are two things that are very important to keep in mind. Is that it has a directionality. The one end is called the plus end, and the other end is actually called the minus end, right? So actually, what plus end means is that in this end the recruitment of new tubulin heterodimers is going on. So here the tubulin dimers are mostly GTP bound, and more new tubulin heterodimers are getting recruited to this end of the microtubule. That means that in this end, the microtubule is elongating, right? This is the elongating end or the polymerizing end or the assembling end of the microtubule. It's keeping on growing because of addition of new heterodimer units to this end of the microtubule. Whereas this minus end typically means that here the GTP that here the GTP, which was initially bound to the tubulin heterodimer, is now getting hydrolyzed to GDP, which is making it more prone to getting disassembled. So this end is the disassembling end of the microtubule, right? And now there is one more significance that when we will look into the polymerization or how the microtubules are formed, then you will find that in a cell, these microtubules are formed from a microtubule organizing center, which are present just around the nucleus, that is they're present perinuclearly. They're present just around the nucleus mostly. So where the microtubules start assembling, and then they grow to the plasma membrane. So basically, this nuclear end of the microtubules is a negative or the minus end, whereas the end of the microtubules towards the plasma membrane are the plus end. This is very important also because of this sense that microtubules act as important highways, right? Important highways for the transport of vesicles having cellular cargo, as well as transport of cellular organelles, right? So there, these motor proteins are very sensitive to this directionality of the microtubule. So only a specific cargoes will be transported by a specific set of motor proteins in the plus oriented manner. That is from the nucleus or from around the nucleus to the plasma membrane. Whereas there will be a specific set of different cargoes which will be transported in this direction, that is from the plasma membrane towards the nucleus by a specific set of motor proteins, right? So this directionality is very important. And that this directionality arises because of this fact that one end there is more GTP hydrolysis to GDP, which makes it more disassembly prone. And this plus end is actually the end where more and more tubulin heterodimers are getting recruited, and the microtubule is growing. So this is the plus end, which is present typically towards the plasma membrane. Right? Okay. So this is what I was mentioning previously, that the microtubules start getting polymerized um, in the microtubule organizing center. So these microtubule organizing centers are basically the centrosomes, the centriole associated proteins, centrosomes, which together are called the microtubule organizing center. This tubulin heterodimers start polymerizing and assembling to form the microtubules, which are further elongated so they start polymerizing. Here you can see the micro MTOC. They're very small. Here is the enlarged view. So the microtubules start forming because of polymerization or assembly of, not polymerization, because of assembly of these tubulin heterodimers just outside the nucleus, that is in the perinuclear region, from which these microtubules grow gradually towards the plasma membrane. Right? So near the plasma membrane, it will be the plus end, and near the nucleus, it will be the minus end. In the green, you can see In the green, you can see the microtubules, okay? And in the red, basically, this is the actin filaments that you can see, which you can see because of staining with phalloidin, that is conjugated with 590. Phalloidin is a protein that is produced by some fungi, which binds specifically actin. So basically, in short, the microtubule organizing center is which is located just outside the nucleus, is a site where the tubulin heterodimers assemble to initiate the formation or nucleate the formation of microtubules. Here is near the nucleus will be the negative end, and gradually as it is expanding towards the plasma membrane, it will be the positive end. Okay, now let's see about the components of the actin microfilaments. So actin microfilaments, again, they are made up of protein subunits. They are made up of actin protein subunits, okay? Now these actin protein subunits, they also are nucleotide binding, but the nucleotide is ATP. Because of tubulin heterodimers, we saw that the alpha tubulin and beta tubulin are binding GTP, guanosine triphosphate as a nucleotide, whereas actin subunits bind adenosine triphosphate as the nucleotide, right? And here also this ATP bound actin subunit polymerizes or basically assembles head to tail manner to form protofilaments. And here what happens is that two actin protofilaments get intertwined, right? Two actin protofilaments get twisted or intertwined to form a long filamentous structure, where these two protofilaments are held together because of lateral contacts, right? So they get intertwisted or intertwined to form a right-handed helix-like fibrillar structure. And interestingly, even these these actin cytoskeletal elements, they provide considerable mechanical strength and rigidity as well as flexibility to the plasma membrane, which are necessary for cellular functions like the maintaining the cell shape, cell migration, cell mobility, the membrane remodeling, right? That is invagination of the cell or protrusion of the cell for phagocytosis, endocytosis, etc. And just like the tubulin or the microtubules, the actin, these are also very dynamic molecules. Like there is also polymerization or assembly and disassembly, right? There is also a balance between assembly and disassembly. And because of that, and they also have a directionality. There is also assembly and disassembly. And depending on which end you are adding more actin subunits, and at which end there is more actin present in the ATP bound state, and you are actually recruiting more actin subunits, therefore you are elongating the actin molecule, versus one end will be disassembling the actin molecule more because there is more hydrolysis of ATP. So in this minus end, you will have more hydrolysis of ATP of the actin subunits. There will be more in the ADP bound state and there will be more disassembly, right? Whereas in the plus end, there will be more actin subunits present in the ATP bound state and there will be more assembly of actin subunits and therefore elongation of the actin fibril or the actin microfilament, right? So that will be the plus end. However, the actin is present primarily here. The actin microfilaments are present just beneath, mostly just beneath the cell surface, right? So here there will be, like, for example, this may be the plus end or minus end, but again, this directionality is present in the actin microfilaments as well. And with actin, there is one interesting characteristic which is quite different from that of microtubules made up of tubulin assemblies. That is, tubulin assemblies which form these microtubules, which are hollow tubular networks, are long unbranched fibrils, right? Whereas actin is capable of forming branchings, which are formed by a which are formed by a set of proteins like ARP2 and ARP3 in a very regulated manner. But actin microfilaments have the capability to form branched microfilaments that lead to a mesh or network beneath the membrane, And this branching, debranching, as well as polymerization, depolymerization helps in govern the remodeling of the actin microfilament, and therefore remodeling of the membrane shape and structure, which are important in terms of endocytosis, these membrane protrusions, cell migration, right, cell motility, cell movement, etc., as well as during this cytokinesis of mitosis or meiosis. And this is a electron microscopic image which shows the mesh or network of branched microfilaments. So you can see that this was one active microfilament, but there is a branching here. It's a very unique property of actin microfilaments as opposed to the tubulin microtubules. So yeah, this is the electron microscopic view that shows you the meshwork or network of branched actin microfilaments in the lamellopodia. That means lamellopodia is this. For example, this is the surface. You have an amoeba cell, right? And the amoeba cell is extending its membrane, or even in like mesenchymal cells of humans, there can be extension or protrusion of the membrane for cell motility or movement in a certain direction. This is the lamellopodia. There you will see growing of actin microfilaments and branching of actin microfilaments for facilitating cell movement, migration in a certain direction. Right. So, okay, this was kind of the summary of big five contents of this course, Introduction to Cell Biology, from my end. If you have any questions, then we can discuss from this week five or even earlier weeks. And if you have any questions from the assignments of week five, you can also ask. Okay, so if there are no questions, I hope that you understood, and I will be able to do your assignment questions and submit them in time. Then bye for today. We will meet in the next session, next Sunday. Bye.